And our next presenter is Marshall Porterfield. And Marshall, are you ready?
I'm ready.
All right, you have the floor.
Good afternoon. So, in an era of convergence, as fields start to coalesce and become new fields, we see the emergence of new fields, also comes with that new vocabulary. So, I'm going to talk about Biosystomics today. I'm going to talk about integrated omics and try to draw a distinction between what's been done in the past and what we see is going to be happening in the future. So our freshman class today at Purdue was born at the dawn of the genomics era. Now, while there were great promises and expectations that were born with this era, I would argue that we haven't really achieved as much as what we expected that we would have achieved having access to the human genome, the code of life. And since that time, there have been thousands of other genomes of other species which have been published. In the early stages of the Human Genome Project, the molecular biologists were having problems dealing with the data. So they teamed up with information science, and what was born was bioinformatics. But we still haven't achieved what we possibly can achieve. And so what we need to do is we need to look at the system, and really this is a system of systems. So this is a very simple diagram that basically takes a look at the genome with epigenetics, the genes, the transcriptome, the proteome, the different components of the proteome, leading to the metabolome. So these are 5 dimensions of data. Out of the genome, about 3% we know code for genes. That's all that we can figure out about the genome, 3%. That's where we're at today. How do we figure out the rest of it? That's the key challenge. Now, I like to use a computer science analogy to explain the problem. So we have the genome, which is equivalent to the hard drive. Somewhere in there also is the operating system and the CPU. Out of the hard drive comes messenger RNA, which are the programs. The proteins are these application programs which give rise to the metabolite, which are basically the I/O, input-output. So what we need to be able to do is look at these different dimensions of data and integrate them in a way where we can create an emulator of what the operating system is for a cell and map that back to the genome itself. So this is, I'm going to show you that this is big data on steroids, and it's a really big system of systems problem. And that's why we coined the term BioSystomics, of trying to integrate these different levels of the flow of biological information into biological activity. So, multi-omics is not integrated omics. Multi-omics, the Cancer Genome Atlas, and other affiliated types of databases, database collections of transcriptomic data, proteomic data, it's all stovepiped by the NIH currently. People are trying to harvest data from these data systems to make new discoveries. Sometimes it's possible, sometimes it's not, using machine learning and artificial intelligence. But the challenge is that the data that populates these publicly open data systems come from experiments from different labs, with different experiments, with different scientific protocols. There's a lot of noise in the system. And the old computer science saying of garbage in, garbage out applies here too. How do you know that your machine learning tool or your artificial intelligence works if you're just using this type of data? You need a calibration standard. You need a model reference system in order to test the artificial intelligence of machine learning tools. Right now, we're kind of fumbling around in the dark using these tools and hoping that the data and the information, the knowledge it's yielding is true, is real. So again, the goal is to create this multi-dimensional omics, the 5 levels: transcriptomics, genomics, epigenetics, proteomics, and metabolomics. Each one of these planes with between 20, so there's 20,000 genes, 6 billion base pairs in the genome, tens of thousands to hundreds of thousands different types of proteins and metabolites. At a conservative level, if you combine all that in terms of the data complexity and you compute that, it's something like 10 to the 19. So this is big data on steroids, also because of the data complexity of the system. So what we need is some kind of integrated reference standard, a calibration to test and develop the AI and machine learning tools. What we were proposing was to look at very simple model systems, experimental versus control. We also wanted to evaluate the variability within an omics channel. So how many times do you have to run a transcriptomic analysis? I've talked to people who say they sometimes see greater differences in their replications than they do their experimental versus control. So how do we get the highest level of precise data into a reference system to develop these tools, which then can lead to the big breakthroughs in discovery? If we can understand the operating system of a cell, if we can reverse engineer it, and we can map it back to the DNA sequences, we can reinvent it too. And so it offers great opportunities for development of medicine and agriculture, all realms of life sciences really in the future. Thank you very much for your attention.
We have time through the transition here for a question or two for Marshall. Okay, no questions.
I guess under all this, there is assumption that you have to be able to deal with an enormous amount of data. So what is, to bring these things together which you have in mind, what is your idea, how to really deal with this amount of data coming from so very different areas?
What you need to basically is create that reference experiment to act as a calibration. Just control versus one factor, and keep it very simple and clean, and that way you can measure, collect all the data from those two treatments and start simple and work with that in order to develop the machine learning and AI tools. So it really comes down to, it's basically, I'm a sensor guy, right? We want to build a BioSystomics sensor. How do you do that if you don't have any calibration standards? How do you make a thermometer if you don't have boiling water and ice? And that's really what we need to do, is create a very simple data system. Now, the implications of this is once you can create these reference data systems, you change the landscape for science management policy. So National Cancer Institute could put together these reference experiments for every type of cancer. That way the community of researchers that are doing cancer research in those areas are sharing from the same pool of observations to develop translational discovery opportunities.
Marshall, thank you so much.
All right.